Thank you very much. Nice to see you all. I want to talk today about three things. I want to just do a very brief introduction to the pharmacogenomics knowledge base that we're building and that associates human genetics with drug response phenotypes. And then I want to talk about the annotation of a complete human genome as kind of going in the direction that Soren just mentioned of our rising ability to sequence humans and then to make inferences about their health. And then I want to say a few things about rare variations in the genome, because one of the differences between genotyping platforms, where you measure mostly common variants, and full genome sequencing is you're going to make many discoveries in the genome sequence that you have no idea what they mean, and yet the patients will care very much because they have these variations. So this is the PharmGKB homepage. I've said many times I never recommend giving a talk about a website, but I'm just show 5 or 6 slides. The thing I want to say about the PharmGKB is it's built by a team with 2 parts. There are PhD and master's level curators who look at the literature and turn the published literature into annotations and knowledge that's stored in the database. And then there's the developers who on the informatics side build the site and build the tools that use the site, that are on the site. I will mention that on the far right and the upper right, the curators pick their favorite papers in pharmacogenomics every 2 weeks. And so for those of you interested in learning more about pharmacogenomics, these are highly selected papers that they think are worth reading. And it's at pharmgkb.org. So the other thing I wanted to say is when we were thinking about search, we actually have these 5 different search boxes, and the semantics of these searches are you can type anything you want into the search box, but the answer will be of the type that is specified. So for example, in the next slide, this is the gene search, and you can type in a disease or a drug or any other words, but the answer is guaranteed to be genes. So in this case, I've typed in warfarin, which is a drug, an anticoagulant, and what you get is this is the first part of the hit list. These are genes that have been annotated as having something to do with warfarin response or coagulation, which is the biological process that warfarin interferes with. If I go to VKORC1, which is a famous gene that modulates warfarin response, you have a list of SNPs that the curators have annotated, which give you detailed information about the different alleles of those SNPs and how they relate to the drug response. So I can go to the variant search and I can type in warfarin, and then I'll get variants in the human genome associated with this drug. And so what you will get here are these little snippets where the curators have said this is a SNP from DB SNP code. It's on chromosome 10, and in this case it says the variant has been shown to influence warfarin dose and several other drugs, and then there's some controlled vocabulary to allow searching and automatic analysis. Now, very soon we will be releasing a new user interface to this data, which we're pretty excited about. This will be for warfarin. This is a list of all the relevant genes that have been curated by our staff. And we will also have a more detailed annotation of the information about the phenotype-genotype association. So here we're saying genotype AA. So this is a SNP with 2 alleles. And if you are homozygous for AA versus AC or CC, it has a certain phenotype, according to this paper that was published. And they actually now are including the study size, the ethnic origin, the p-values, and this is all kind of in a standardized format, which will hopefully help data mining applications and discovery. And then the third new thing that we're going to integrate is we're going to take all of those variants and we're going to turn them into a clinical summary, which is if you are a person who has an AA at this particular SNP, then you have an average chance of responding to warfarin. You have an average, that's not worded very well, but you have an average expected response, whereas for different genotypes, you might need a higher dose or a lower dose or have a higher risk of side effects. So these are the best effort of our curators to take all the literature on a particular SNP and integrate it into a clinical interpretation, which involves obviously some medical expertise. The reason we do this is because we are headed towards a time when we will be looking at genomes, genotypes, and whole genomes, and we will want to give people reports, or their physicians, reports of what is expected. That's coming out soon. The last 2 or 3 slides that I showed you are in our beta site, but not yet on our production site. And then, of course, warfarin is a drug, and if you search for drugs, you will get the warfarin page, and you will get information about the drug. And then pathways, and you will get, we have these pathways that are created by our curators. These are very expensive for me because they're laid out by hand. Every single object on this network has been curated, and all the links have been curated. You can actually download the supporting references that support this. And I think this creates, we have about 60 of these right now. They come out slowly, but I think they're very high quality kind of systems pharmacology networks that are fairly well vetted and documented if you need kind of gold standard drug-related pathways, both for pharmacogenetics, how the drug is metabolized, as well as pharmacodynamics, how the drug actually works. Okay, so about 6 months ago, we published a paper where we took the entire contents of pharmacogb and applied it to a human genome. My colleague Steve Quake sequenced his own genome about 2 years ago. He wrote a paper that was in Nature Biotech, which just the details of how he did it technically. But he came to us and he said, well, what does this mean for my health? And so we were treating him as a patient. By the way, he has agreed to have his identity released, so it's okay for me to mention his name. In fact, it was on the cover of the New York Times when he published his own genome, so he had no interest in privacy, evidently. He was not on any medications at the time. In his genome, he had about 3 million SNPs, about 2.1 million of which were well described already. They had been seen before. But 370,000 of these SNPs had never been seen before in a human. Now, there hadn't been that many humans sequenced, so these are not all rare, but they were all novel to us at the time. He also found some copy number variations, which I won't discuss. So last year, we published this clinical assessment of his genome. We broke it into 3 teams. One team did Mendelian disease risk. Another team did common disease risk. That was led by Atul Butte and Ewan Ashley. But my team did the drug response, and that's really what I'm going to focus on. So we had about 2,500 SNPs that we looked at that were common, known variants. We also looked at the copy number variants in our VIP genes. PharmGKB has this thing called very important pharmacogenes, which are genes that are frequently implicated in drug response. And then we looked at the novel SNPs only in those VIP genes, because in only those genes would we be able to predict what might be the effect of these novel SNPs. So a couple of the highlights are that the patient was heterozygous for a null mutation of a very important metabolizing enzyme, CYP2C19. This so he has one normal copy and one dysfunctional, non-functional copy. And this has been described. This is a common variant. It's been studied. And so we know that he will have an altered metabolism of a number of drugs, including proton pump inhibitors, which are antacids, anti-epileptics, and then a potpourri of other drugs, including clopidogrel, which is an anticoagulant where the United States Food and Drug Administration has recently changed the drug label, saying that patients with variations in this gene in particular might want to stay away from that drug because it is highly variable in its response in the context of this variation. Now, I should say that in 2009, when he sequenced his genome, it cost him $48,000. So the one question is this information worth $48,000? And I think the answer is no. I'm pretty confident the answer is no. Although if he took clopidogrel and got into trouble clinically, it could easily run up a medical bill of $48,000. However, many of you know that genomes are now less than $10,000, in some cases less than $5,000, and then you're getting perilously close to having it be cost effective. Now, I just want to summarize 3 tables. Pharmacogenetic bad news. which I'm sorry you may not be able to read, but these are drugs for which he would be expected to have a higher chance versus the normal population, not normal population, but the average population, higher chance of side effects, higher chance of having the drug not work, or more likely to have a difficult to predict dose. And so, for example, both warfarin, the anticoagulant, and clopidogrel, which I just mentioned, are both problematic. So if he ever needs to be anticoagulated because of a clotting problem, he will be a challenge for his physician, is the prediction here. So again, is that worth $48,000? Maybe. Is it worth a few thousand or a few $100s? Definitely. And then there are other drugs here for diabetes. At the very bottom, sildenafil or Viagra is less likely to work based on some studies. Now I should say that you see the level of evidence here is based on how big or how many studies were done for this information. And I think it's very important to stress that high evidence, which is 2 independent studies, is pretty rare. Medium evidence is pretty rare, and most of these studies are what we characterized as lower evidence. Lower evidence would be a single study of less than 150 or 100 patients, and so it could be wrong, but it could be right. And so that's really where most of the literature is right now. Good news is the opposite. Things that are more likely to work than expected in the population, less likely to have side effects than expected, or more likely to have a stable dose. And so this is the good news, and one of the pieces of good news is he can drink caffeine relatively free from worry of heart palpitations. He is likely to benefit from a cholesterol drug if he needs to lower his cholesterol. He does not have the risk factors for some of the side effects associated, and it goes on. And then really is the most interesting table, I think, and the one that gives bioinformaticians plenty to do in the next 10 or 20 years, which are the novel SNPs. So this is just a table of the novel SNPs that were in our VIP genes. So these are genes that are known to be important for some drugs. Drugs are listed on the right, which change an amino acid in a way that appears to be deleterious, using SIFT or other programs like this that look at the nature of the amino acid change to make a prediction. So these are pretty big changes in the proteins. Sometimes they're even nonsense, either changing an amino acid or truncating a protein. So what do we tell the patient? Well, the first thing we say to the patient and their doctor is, we have no idea. We have never seen this variant before, period. Pause. Then the next thing we can say is, but this gene is important for these drugs, or each row is a single gene. And if you are about to use this drug, you may have an unexpected response because you have uncharacterized variations in a gene that is very important for that response. So while that is not precise advice, in many clinical settings, that could be very useful advice, especially if you're in an area where there are many choices. And with this information, you might say, I'm going to go to another class of drugs because there's some question in this class. So this will play out over time. I think we can do, with the kind of work that Soren just described, we will be able to follow patients who have these interventions, and we will be able to determine if it's working or not as we use it in clinical practice. So obviously there are limitations to this analysis. We only looked at the VIP genes for many of the variations because those are the only ones we knew how to interpret. So there's a huge opportunity for systems views of pharmacology, which I'm a big fan of. As I pointed out, the level of evidence for many of these clinical inferences was low. We are always worried about errors in sequence. We didn't look at non-coding regions at all, and right now the patient has no diseases and not taking any drugs, so this is all looking towards the future. So my intermediate conclusion is that the PharmGKB provides a useful summary of current knowledge that can be used to annotate a genome even today. It's an imperfect annotation, but it gives you a flavor, I think, for what we're all headed for. And then I won't talk about this, but I recently published an editorial in Clinical Pharmacology and Therapeutics arguing that pharmacogenomics is ready now for a limited implementation where we instrument the system and watch very carefully, but as long as we're sure that we're not likely to hurt the patients, we can probably go forward to see if we're actually helping with prospective trials. Now, I just want to tell a little story, a little bit of detail. I think I'm okay for time. 5 minutes, perfect. So warfarin I've mentioned before, or Coumadin, it's a blood thinner. There are two main genes involved in the response to warfarin, a cytochrome 2C9 and a pharmacodynamic gene VKORC1. And over many years, people have figured out how to use variations in these genes to actually predict the dose of warfarin. Warfarin has a very variable dose from 2 milligrams a day to 20 milligrams a day, and it's very difficult to predict because you can't predict genetics very easily. There are some associations with ethnic origin, but they're not very strong. So a couple of years ago, we were part of a big international consortium that published a pharmacogenetic algorithm that said, here's how you can predict warfarin dose using genetics, and we showed that it far outperformed an algorithm using only clinical data. That is to say, including the SNPs, increased the R-squared of the ability to predict greatly. And in fact, this is a plot of the observed dose of warfarin on the y-axis versus the predicted dose on the x-axis, and this was a pretty good correlation with about 52% of variation explained by the genetics combined with clinical data. Then we were interested in extending this for rare variants. One of the things we did was we looked at 2C9. In the original equation, we only looked at common variants. We went into Stanford. I wanted to get to this because I wanted to get to electronic medical records, so thankfully I'm here. I could stop right now. So we have a warfarin clinic at Stanford, and we were able to enroll 104 patients where we had their full phenotypic data from their electronic medical record, and we also genotyped them, not just at the common locations, but at some rare locations. And what we found is if we pooled all of the rare variants of CYP2C9, in fact, there's 8 additional variants that we used, we were able to greatly improve the predictive ability of the algorithm. So why is this interesting? We couldn't add them as single variables because we didn't have enough data and it would be too noisy. However, if we just pooled them and made the somewhat risky assumption that they were all loss of function mutations, which is of course not guaranteed, but in this case, it wound up improving our R-squared by as much as 12%. So we're very happy about that, and it showed a way towards perhaps even including rare variants in clinical decision-making. That was the high-level point here, is we're going to have to figure out when somebody comes to us with a rare variant in their genome, how to use it. We're also interested in the African-American population who are not well served by our current algorithms. Really, only 53% of African Americans have variations that are found in Caucasians and Asians, and therefore the algorithm's R-squared is much worse for this population. And in fact, if you look, the average dose of somebody of African origin of warfarin is higher than those of Asian or European origin. So we're doing a couple of things just to close up. We're doing a GWAS focused on African Americans, trying to find new common variations that might explain the dose requirements. And we're also sequencing 50 high-dose and low-dose exomes, hoping to perhaps find rare variants. And we are thinking very hard about the methods with which we'll be able to interpret these rare variants. Just to jump ahead and finish up, we have these methods based on collapsing, which you may have heard of, where you look at a gene and you look at the distribution of minor alleles across the entire gene. Again, because they're rare, you can't do statistics on any individual, but you can any individual variation, but you can group them together. So, for example, in a measure that we call p-Hirsch, because a guy named Hirsch invented it in our lab, we just count up the number of minor alleles in patient 1 and patient 2. Patient 1 has 3 minor alleles, shown in red. Patient 2 has 5. We can consider this basically a quantitative trait, and we can correlate that to a phenotype of interest. And in fact, when we do this in our initial GWAS data, we see that this p-Hirsch, or there's a variation of it called p-Hap score, correlates with dose. That is, the burden of minor alleles in the gene correlates with the dose of warfarin. We don't know exactly which variations because we've collapsed them all together, but we see a pretty strong correlation. And in this case, this gene looks pretty good. It's involved in platelet dysfunction, and platelet dysfunction is involved in clotting. So the story looks like it may be, very early, may be true. So I will stop there and conclude that pharmacogenomics is a great test bed for whole genome applications, including annotating the literature, bringing it to clinical practice. Rare variants are important and continue to be very challenging. That's where our head is really for the future. We have some methods that show promise, and we are excited about the opportunities for informatics to make contributions in this area. So I think I will stop. I'm a member of ISCB, and I want to make sure I say that because I think we all need to be proud of our membership in this organization. And I do want to give a plug for a translational bioinformatics meeting that is held jointly between ISCB and AMIA each year. And, of course, my co-workers and funding. Thank you.